Hello and welcome to another video by Perfect Scores. This is Prenatal, and what we're going to cover in this video today is a very useful process known as karyotyping. Now in the previous video we discussed about meiosis and what is non-disjunction. That means when the chromosomes they fail to separate. So how do you know that the chromosomes have failed to separate, or whether the person has an extra chromosome or there is a difference in the number of chromosomes? And how do we answer all those questions? The answer is given by this technique known as karyotyping. So what exactly is a karyotype? A karyotype is nothing but a visual profile of all the chromosomes in a cell. So visual profile of all the chromosomes in a cell. So what is basically done is all the chromosomes, they are arranged into homologous pairs and then they are displayed according to their structural characteristics. So this diagram that you can see, that is example of a normal karyotype of a human. So you can see it has 1, 2, 3, 4, 5. Like that it has all the 22 pairs, and the last pair, which is the 23rd pair, that is known as the sex chromosome pair. So that's why it's showing X versus Y. And the remaining 22, they are known as autosomes because they do not depend on the gender. Now, if the genes or if the chromosomes are X chromosome and another X chromosome, that gives rise to a female zygote. But if there is X chromosome and a Y chromosome, that gives rise to a male zygote. So basically over here, let's suppose a person has the Down syndrome. That means at number 21, you will be able to see one more extra chromosome. That means there are going to be a total of 3 chromosomes of number 21. So that will be the kind of trisomy that is there. So the next important thing is, how is actually karyotyping done? So we are going to study about prenatal karyotyping now. Prenatal karyotyping means karyotyping done for a fetus that is yet to be born. So that's why we call it prenatal karyotyping. That is done in order to determine any possibility of a genetic disorder before the birth of a child. So basically it is used for two things. One is to determine the gender of an unborn child. The other one is to test for any kind of chromosomal abnormality. So there are basically two methods that you need to know. One is amniocentesis. So in amniocentesis, a needle is inserted through the abdominal wall and the cells, they are taken from the amniotic fluid which is present in the amniotic sac around the fetus. And this amniotic fluid contains the fetal cells that can be taken, and this is usually done at the 16th week of pregnancy with a slight chance of miscarriage, which is about 0.5%. So 0.5% chance of a miscarriage. The second technique is the chorionic villi sampling or the chorionic villus sampling. So in this case, a tube is inserted through the cervix and a tiny sample of the chorionic villi from the placenta. Now these are finger-like projections in the placenta. So cells from the placenta are taken. That can be done at the 11th week of pregnancy, and it has again a slight risk of inducing miscarriage, which is approximately 1%. So let's go ahead and now analyze what all is there under the human karyotype. Now in the human karyotype, what we observe is that every cell in our body has 46 chromosomes. That means 23 pairs. That means 23 from the mother and 23 from the father. And I'm talking about every cell, but there are exceptions. For example, haploid gametes. They don't have 46 chromosomes, they have 23, and red blood cells who do not have a nucleus at all. Now, the males and the females can be differentiated on the basis of their sex chromosomes. Additionally, the non-disjunction that can lead to individuals with an abnormal number of chromosomes, so we can talk about that as well. So 23 pairs, it gives us an idea of gender, and it gives us an idea of any kind of genetic abnormality. And lots of disorders are there that can be classified according to the number of chromosomes that are affected and the number of chromosomes that are present. So that is all that we will be covering in this video, which is based on karyotyping. The next video is going to focus more on theoretical genetics, in which we are going to discuss lots of concepts such as genotype, phenotype, dominant and recessive alleles, and so on. So thank you so much for watching this video.